السلام عليكم ورحمة الله. معكم الدكتور محمد نجيب عمر, Professor of Medical Biochemistry and the Faculty of Medicine, Jazan University, and Faculty of Ain Shams University in Egypt, Cairo. My lecture today is about the extracellular matrix, the biochemistry of the extracellular matrix as a matter of fact. By the end of this lecture, we will be able to understand the composition and function of extracellular matrix, what are proteoglycans and glycosaminoglycans, the differences between them, and the biochemistry of collagen and other extracellular proteins. And lastly, we are going to discuss the disorders associated with abnormality in extracellular proteins. Let us start by definition of the extracellular matrix. The extracellular matrix is a collection of extracellular molecules which are synthesized and secreted by the different cells. It is actually the ground substance that provides structural and biochemical support to the surrounding cells. It is important for cell adhesion, cell-to-cell communication, and differentiation. Now, what are the major molecules that form the extracellular matrix? These are the glycosaminoglycans and the proteoglycans, and we are going to understand the difference. Collagen, elastin, and a group of glycoproteins termed adhesive glycoproteins. These adhesive glycoproteins include laminin, fibronectin, integrins, and fibrillin. These are the major molecules forming the extracellular matrix: proteoglycans, fibronectin, collagen, laminin, etc. We are going to start by glycosaminoglycans and proteoglycans. Glycosaminoglycans are linear carbohydrate chains of repeated disaccharide units. So the glycosaminoglycans are characterized by being formed from repeated disaccharide units, and they form linear chains, linear carbohydrate chains. There is no branching. One unit of these carbohydrate chains is always hexosamine, such as glucosamine or galactosamine. The other unit is usually uronic acid molecule, mainly glucuronic acid and sometimes L-iduronic acid. What are the types of glycosaminoglycans? There are 6 types of glycosaminoglycans. Hyaluronic acid, which is present in the synovial fluid of joints, and it absorbs shock, so it is shock absorbent. Dermatan sulfate, present in the skin, blood vessels, and the heart valves. Chondroitin sulfate, present in cartilage. Heparin, present in the form of granules of mast cells, and here we observe that heparin is the only extracellular, the only intracellular glycosaminoglycans that is present inside the mast cells. Then it is secreted or released from the mast cells. Heparan sulfate, it is present in the basement membranes and all cell surfaces. Keratan sulfate, which is present in cornea and conjunctiva. What are the general characters of glycosaminoglycans? All glycosaminoglycans are negatively charged molecules. Why? Due to the presence of sulfate, as we observe that sulfate is present in all types of glycosaminoglycans, with only one exception, which is hyaluronic acid. So all glycosaminoglycans contain sulfate. That's why they are all negatively charged. In addition to the sulfate, they contain carboxylate groups of uronic acid. So the negative charges on the sulfate and on the carboxylate groups forming the glycosaminoglycans render such molecules highly negatively charged molecules. Number two, all glycosaminoglycans are highly charged polar hydrophilic molecules, bind water and become highly viscous. Because of the polarity and the charge of the glycosaminoglycan molecules, they bind water and become viscous in the solution. All glycosaminoglycans contain sulfate groups and are found covalently attached to proteins with one exception, which is hyaluronic acid. So hyaluronic acid is negatively charged because of the carboxylate groups of the glucuronic acid or the uronic acid enters in its structure. But hyaluronic acid is not having sulfate group and it is not covalently attached to proteins. What is the clinical importance of glycosaminoglycans? Heparin, as we all know, is a glycosaminoglycan which is important as anticoagulant. It is found in the granules of mast cells. Heparin activates the antithrombin 3 enzyme, which inactivates factor 2A thrombin, factor 9A, and factor 10A. So heparin increases the antithrombin 3 activity by 1,000 folds. It can be used during the treatment of myocardial infarction. So the clinical importance of one of the glycosaminoglycans, which is heparin, it acts as anticoagulant through activation of antithrombin 3, which inactivates thrombin, factor 9A and factor 10A. In addition to the other functions of glycosaminoglycans as those present in the joints and the vitreous humor, under the skin, etc., and the cornea, etc. Then, what are proteoglycans? Proteoglycans are nothing but glycosaminoglycans covalently attached to proteins. So, glycosaminoglycans plus protein equals to proteoglycans. This is a proteoglycan. It is formed from a protein, and this protein attached to it the different types of glycosaminoglycans chain. Because the glycosaminoglycans are highly negatively charged, they will repel each other and present separated from each other in the glycosaminoglycan molecule. The glycosaminoglycan monomers then form the proteoglycans monomers, which is formed from a core protein to which is attached the glycosaminoglycans formed of repeated disaccharide units and negatively charged. As I said, these proteoglycan monomers then they will attach to a molecule of hyaluronic acid to form the proteoglycan aggregates. So the proteoglycan aggregates are formed from proteoglycan monomers that lie and attach by a link protein to the surface of hyaluronic acid. So hyaluronic acid attaches to these monomers to form the proteoglycan aggregates. What is mucopolysaccharidosis? Mucopolysaccharidosis are a group of genetic disorders. They result from deficiency of lysosomal enzymes normally involved in the process of degradation of the glycosaminoglycan chains of proteoglycans. So the proteoglycans are engulfed and get to the intracellular fluid. then they are degraded by means of lysosomal enzymes that degrade the disaccharide units of the glycosaminoglycans. If such enzymes, lysosomal enzymes, are deficient, then mucopolysaccharidosis, a disease, will arise. Mucopolysaccaridosis are manifested by accumulation of proteoglycans inside cellular lysosomes with degeneration and the death of the cell. This results in mental retardation, skeletal deformities, and early death. And this is some pictures of some patients suffering from mucopolysaccharidosis. Second, let us discuss collagen. Collagen is another member of the extracellular matrix. Collagen is widely distributed in our body. Collagen is a fibrous protein. It makes about 1/3 of the total protein mass of our body. So it is widely distributed in our body. Collagen is present everywhere, in the skin and the tendons and the ligaments and the bones and in connective tissues. Concerning the structure of collagen, collagen is formed from tropocollagen molecules. The tropocollagen molecules are the basic structural unit of collagen. It is formed from 3 super twisted polypeptide chains. They form a triple helix. So the 3 polypeptide chains are wrapped or twisted around each other to form the tropocollagen. The tropocollagens then form the collagen fibers and the collagen fibril and then the collagen fibers. This triple helix, which is the basic unit of collagen, is formed from repeated amino acids. Every third amino acid is glycine. So the polypeptide chains of collagen are rich in glycine, as glycine will represent about 1/3 or 35% of collagen amino acids. It is repeated every third amino acid in the polypeptide chain. So every third amino acid will find glycine. Why glycine? Because glycine is the smallest amino acid. So the polypeptide chain will be formed of glycine XY. XY means other types of amino acids. Collagen, because of that, is poor in many essential amino acids. Thus, it is a protein of low biological value. Collagen is degraded by the enzyme collagenase enzyme and it fragments collagen into small pieces. That's why the signs of aging starts to appear as we grow in age. Collagen production naturally decreases by approximately 1% each year. This occurs due to its degradation by collagenase enzyme. This loss is accelerated by sun exposure and hormonal changes during the process of menopause or during the age of menopause. This results in reduction in the normal volume and development of wrinkles. As we see, by age, there will be wrinkles due to destruction of the collagen by the collagenase enzyme. Inhibitors of collagenase may protect against such changes, and many of the creams in the market are claiming that, and many of the beauty creams claiming that they protect collagen from destruction, or they may add collagen to the skin and rendering the skin more younger in age. Now let us start by discussing some of the disorders that may affect collagen. One of the diseases that may affect collagen is scurvy. And scurvy is a dietetic disease. It results from deficiency of vitamin C. Vitamin C is required for the activity of the hydroxylase enzyme, which is essential for hydroxylation of lysine and proline amino acids present in the polypeptide chains of collagen. Such hydroxylation is responsible for collagen strength. Deficiency of vitamin C will lead to defective hydroxylation of proline and lysine of collagen molecules, giving weak collagen. Weak collagen gives weak connective tissues and the appearance of the scurvy. In scurvy, there is weak connective tissue, so there will be bleeding from the gums, loose teeth, easily fractured bone, and delayed healing of wounds. These are the manifestations of scurvy. So, vitamin C is important for the activity of the hydroxylase enzyme for converting lysine and proline to hydroxylysine and hydroxyproline, essential for glycosylation followed by glycosylation, and such hydroxylation followed by glycosylation is important for the strength of collagen. Here is a picture for the clinical manifestations of scurvy, in which there is weak, fragile, and bleeding gums with weak, loose teeth. Another disease which may affect collagen is osteogenesis imperfecta. This is a genetic disease in comparison with scurvy. So scurvy is due to vitamin C deficiency, while osteogenesis imperfecta is a hereditary congenital disease in which there is gene mutation with replacement of glycine by bulky amino acids such as tryptophan and branched chain amino acids. Glycine, every third amino acid, is important for the super twisting or super coiling of the triple helix of collagen. If glycine is replaced by bulky or branched chain amino acids, then the super twisting of the triple helix will be very weak, and consequently collagen will become weak. Replacement of glycine will affect triple helix formation. Collagen will become weak. Weakness of collagen will cause bone and skeletal deformities. Such disease demonstrates how glycine is important for collagen stability and structure. A third disease of collagen, which is also a genetic disease, is Ehlers-Danlos syndrome, in which there is genetic defect in collagen and connective tissue synthesis and structure. In Ehlers-Danlos syndrome can affect the skin, joints, and the blood vessels. Clinically, it is present in the form of hyper-flexible joints, fragile hyper-elastic skin, arterial rupture, and visceral perforation, with possible life-threatening consequences. Treatment: there is no treatment, only supportive therapy in patients with Ehlers-Danlos syndrome. A third type of extracellular matrix molecules is elastin. Elastins are synthesized by fibroblasts and by smooth muscle. They formed from cross-linked peptides. The cross-links occur at lysine residues. So the elastin, despite the triple helix of collagen, no, it is formed from cross-linking polypeptide chains. The cross-linking occurs at the site of lysine residues present in such polypeptide chains, giving elasticity. So the reason for such elasticity of elastin is the crossing by lysine residues, allowing them to stretch when needed and then return to their original size or original state. This is elastin, then it can be stretched and it can be relaxed back to its original state. Tissues that contain large amount of elastin are the blood vessels, the lungs, and skin. Elastase is an enzyme that degrades elastin, as its name explains or denotes. Alpha 1 antitrypsin is a plasma protein normally secreted by the liver cells into the serum and inhibit elastase. Alpha 1 antitrypsin deficiency leads to uninhibited elastase and destruction of elastic fibers, mainly that present in the lungs, causing destruction of the lung tissues and development of pulmonary emphysema. In this table, we are going to compare between collagen and elastin. The types, collagen, there are many different types, while elastin is only one genetic type of elastin. Stretch capacity is not present in collagen, while it is present in elastin due to the presence of cross links between lysine residues. The primary structure of collagen, it is formed from repeated glycine XY sequences. No such repeating sequence is present in elastin. Triple helix formation is present in collagen while it is absent in elastin. Hydroxylation of lysine or hydroxylysine and hydroxyproline are present in collagen but absent in elastin. Then the last group of molecules or structures that form the extracellular matrix are the adhesive glycoproteins. Adhesive glycoproteins because they adhere or attach to different extracellular matrix proteins. These adhesive glycoproteins include laminin, fibronectin, integrins, and fibrillin. Laminin or laminins are major proteins in the basal lamina, one of the layers of the basement membrane. It has a large molecular weight. It is a trimeric protein formed from three different polypeptide chains: an alpha chain, a beta chain, and a gamma chain. It is formed of three polypeptide chains: alpha, beta, and gamma. Alpha, beta 1, beta 2, or beta and gamma polypeptide chains. Laminin has binding sites for collagen. Here is the binding sites. It can bind with collagen, heparin, and integrins on the cell surfaces. That's why it is one of the adhesive glycoproteins. The second type of adhesive glycoproteins is fibronectin. Fibronectin, a large glycoprotein molecule. It is a dimer formed from two identical very large subunits joined by two disulfide bonds near to their carboxyl terminals. Each chain is having several functional sites. Every chain is having different or several functional sites. Each site is specialized for binding to a particular molecule such as collagen, fibrin, heparin sulfate, and membrane sparing receptor proteins called integrins. So they contain a different adhesive site. That's why fibronectin is a molecule, one of the molecules termed adhesive glycoproteins. Integrins, also known as the molecular glue of life. They are large transmembrane. Here is the plasma membrane, and here is the integrins, large proteins that band through or pass through the plasma membranes. They are large transmembrane glycoproteins formed from alpha and beta subunits, formed from two subunits. They allow cells to attach to the extracellular constituents. Inside the cells, they attach to the cytoskeleton and other molecules, tyrosine kinases, and trigger cell signaling cascades. In the outer side, they attach to other extracellular matrix protein, and inside they can trigger intracellular signaling process. Fibrillin is another large adhesive glycoprotein, a large glycoprotein secreted by fibroblasts. It acts like a scaffold around which elastin fibers are deposited. There is a syndrome associated with fibrillin abnormalities. It is Marfan syndrome. In Marfan syndrome, it is an inherited disease, results from misfolding in fibrillin. The clinical presentation of Marfan syndrome is disproportionately long arms, legs, fingers, and toes, long narrow face, curved spine, and defects of aorta and heart valves, eyes, lungs, and etc. This is the clinical manifestations as we can see in Marfan syndrome. And now, by the ending of such lecture, let us compare between the carbohydrates of proteoglycans and glycoproteins. What's the difference between proteoglycans and glycoproteins? Proteoglycans are long non-branched carbohydrate chains. The carbohydrates of proteoglycans are long non-branched carbohydrate chains, while in glycoproteins the short and branched chains. In proteoglycans, the carbohydrates are formed from repeated disaccharide units, while in glycoproteins they are different sugar units. In proteoglycans, the carbohydrates are highly negatively charged carbohydrates due to the presence of sulfate and the presence of uric acid in the carbohydrate chain, while in the glycoproteins may be charged or non-charged according to the type of the carbohydrate present. That we come to the end of our lecture. Thank you very much.